Welcome to Inside Bioprocessing, a Thermo Fisher Scientific podcast discussing key trends and topics within the bioprocessing industry. In this episode, we'll be focusing on the future of microbial bioproduction. For many years, Escherichia coli, or E. coli, has played a vital role in the manufacture of many life-saving biopharmaceuticals. However, despite this success, the options for E. coli growth media have traditionally been limited compared to eukaryotic production systems. Specifically, there has been a long-standing lack of a chemically defined medium formulated for E. coli. To address this, in 2021, Thermo Fisher Scientific released the first commercially available E. coli fermentation production medium, Gibco Bacto CD Supreme. Today, we're going to speak to three members of the Thermo Fisher team behind this innovation. Please join me in welcoming Senior Research and Development Manager Stacey Holdred, Staff Scientist Neelanjan Sengupta, and Global Product Manager for Gibco Vaccine Media, Grant Martin. So Stacey, could you start by giving us a bit of background to the chemically defined media trend that we've seen over recent years, particularly when it comes to mammalian cell-based bioprocessing?
Sure. So there has been a real evolution over the years in the composition of media, especially for media designed for bioproduction applications using CHO cells. So in the early days of the industry, most of the media contained serum, which was really helpful in getting the cells to grow and to produce the therapeutic proteins. However, as the industry evolved and safety concerns were highlighted with the use of serum, the industry moved to wanting to remove first the serum, then animal origin components. And peptones served a very important role in that evolution because they allowed the removal of serum while contributing to the very positive improvements in cell growth and overall performance, the yield improvements. And so that was really important in the evolution of the industry to get us to where we were able to get good titers of protein using CHO cells. However, as the industry continued to develop, it was determined that we wanted to start going further in the design space to create these chemically defined media so that we could really understand every single component in a formulation and how it was impacting the overall CHO cell performance. And so over the last decade, especially, we have seen a real move in CHO production from using peptones. Not that peptones aren't still used, but we're also seeing a lot of the industry moving to completely chemically defined processes just so that the overall performance of the cultures are being maintained while having good control over what is being used within the process to achieve these yields.
I think you mentioned that it's been a good decade for the development of CD media for CHO cell bioprocessing. So why do you think this trend has only recently reached the microbial bioproduction space?
So that's a great question. When you think about the composition of microbial media, it is typically made primarily of peptones with some additional components added, unlike with mammalian media where there's this really complex medium where peptones are added in a smaller proportion to the overall composition. So with microbial media, the growth was very good with the peptones and continues to be today, by the way. But we were able to still achieve what we were looking to with the peptone-containing media without needing to invest the time and resources to go to a more chemically defined formulation. However, as there is more of a focus in microbial cultures and as this industry is expanding beyond looking at the CHO system and mammalian culture systems, there is a new focus on better defining the microbial media, just like there was with the CHO media. So it is following a similar trend that the CHO culture media did, where, well, we're getting these really good outcomes using the peptones, but now that we are looking at these new application areas, there is a new benefit to maybe better defining the media. And that's where there is now this new trend in microbial cultures to go to more chemically defined formats, just like there was with the mammalian systems.
We mentioned at the start of this podcast that Thermo Fisher has recently released the first commercially available chemically defined microbial media product. Neil, I wondered if you could talk about some of the main challenges that you found during its development.
So one of the main challenges I would say while developing an CD microbial media, and granted, people have tried that in the past, and our focus was on E. coli cultures. So as Stacy was mentioning, peptones offer a lot of advantages. They have larger production. So we were trying to match those targets. So um, matching a nutritionally rich component like peptone could be challenging. The other issues people do run into when they try to develop their in-house medias is they can end up with excessive lag phase, which is the cells are not growing as fast as you would like them to. So what we did was using our unique strategy. We analyze peptones, use those learnings, and develop those into our media solution. So we were targeting peptone performance as our gold standard, use the peptones as learning tool, and incorporate those learnings into developing these CD media.
And has overcoming these challenges impacted the performance of the medium at all compared to non chemically defined alternatives?
We were not compromising on performance because going back to the CHO cell space where people were transitioning into CD media, so performance has to be maintained. So while developing the CD media, we were trying to have a target that it should be equivalent to peptone containing media or classic medias for sure, because that would be a biggest advantage to transition into CD because it will provide consistency, but at the same time, the performance has to be maintained.
Given the long standing success of traditional peptone containing E. coli media, why would biopharmaceutical manufacturers be looking to transition to chemically defined media instead?
Well, I think it's important that everybody has as many options that are available for media as possible so they can determine what works best with their cell line and in their process. So, peptone containing media are still a very important part of this industry, and they drive a lot of very good performance. And so, for those customers who peptone media is working fine, I think that we would encourage them to continue to use that. However, for some customers, they are doing more uh, processing that would need uh, better defined media. And so, in those situations, uh, they might find that peptone containing media don't work as well for them, where they're trying to maybe increase their titers and they aren't able to get there with peptones, or they're having some other internal concerns with using peptones, in which case, having the chemically defined media available that perform equivalently to the peptone containing media is a great option for them because it allows them to increase their performance. It allows them to understand every single component that's within the formulation and still get those really high titers or positive outcomes that they're looking for. And so that's why we thought it was really important to bring a chemically defined CDE coli medium and make it available to everyone, because there really is the need for end users to have a wide variety of options so they can pick what works best for them in their process that allows them to meet their end goals.
And are there any specific advantages that using a chemically defined medium can provide compared to non chemically defined media?
So there are a number of positive attributes to using chemically defined media. So first of all, it can reduce lot to lot performance variability that some customers experience using peptone or less defined media, because we do know the composition of every component that is going in there. Using chemically defined media can reduce or eliminate the risk of bacteriophage and BSE TSE contaminations because we would be using animal origin free components, and we know everything that's going into there. Again, I've already mentioned that we can optimize your fermentation workflow because having this chemically defined formulation, the cells readily adapt to it, and you can just put it right into your process, and you get the positive performance using it as you would with a peptone-containing media. We've already discussed how the performance is equivalent to the peptone-containing media with a wide variety of E. coli strains. One of the things about the chemically defined formulations also is that they also provide you the flexible sterilization methods, just like with the peptone-containing medium, where you can filter sterilize them or autoclave them. So you don't lose any of those advantages by going chemically defined that you already have with peptone-containing medium.
Grant, is there anything else you'd like to add on this point?
Yeah, no, those are all great points brought up by Stacy. Yeah, I think I just want to reiterate, what we hear from customers in their process is that lot to lot performance, consistency, and the impact of variability is huge, right? And that, really the industry was calling for a chemically defined microbial media to meet this need. Certainly along those lines, we see that the elimination of the risk of bacteriophage and viral contamination is very important as well. And having those flexible sterilization options are critical, some of which may or may not be able to reduce some of those bacteriophage or viral contamination. So having the ability to have a chemically defined media that can withstand the pressures of autoclaving, I think, is essential. And then, kind of to close that thought out, being sure that you have a scalable format and a partner that can scale with your needs, like Thermo Fisher, is critical.
So I think we've covered a lot of ground here, having spoken about the past and present of microbial bioprocessing. But in terms of the future, what are the emerging applications of E. coli within biopharmaceutical production that could be enhanced by the use of chemically defined media?
For sure, and that's a great question. Certainly with the past 18 months, we've seen significant growth and interest from the mRNA and DNA vaccine users and market. This fits very nicely in that upstream process. But also, prior to that, with the peptone products offered by Gibco, we saw the plasmid industry supporting cell and gene therapies really starting to take off. So I think if you look at those three items, mRNA, DNA vaccines, and gene therapies, there's a lot of potential and a lot of growth that we're going to be seeing here over the next few years. That's going to be quite interesting.
And lastly, it would be great to touch on what kind of impact this could have on the bioprocessing industry going forward. Grant, do you see this becoming the industry standard for microbial bioproduction? And if so, what time frame could we expect this transition to happen?
Sure, that's another great question. I think as technologies and our products specifically move closer to the patient and the manufacturing processes, specifically thinking about vaccines and gene therapies, I think we hear a lot that consistency, purity, and safety are extremely important. I think offering a chemically defined product in the microbial space supports those objectives, and the full benefits of this market shift really have not been fully realized. I think it's going to take a lot of user feedback and development, but I think ultimately the advantage that our customers may see could be revolutionary. It's going to be very exciting to witness. We're certainly hearing a lot of positive feedback around the product and what it means for the microbial space. We've heard that a lot of time and energy is spent to formulate in-house chemically defined media if users are going down that route. But having this off-the-shelf option for customers is going to be very helpful. It's going to help streamline some of those development efforts and hopefully make it a much more streamlined process through that development through the clinic. In terms of the time frame to expect a change in this market dynamic, I don't believe it will happen overnight, much to Stacy's point with the mammalian space. But as more biotherapeutic and vaccines use this technology and start to report the benefits, I think it'll have a compounding effect. The switch to chemically defined media in the mammalian space took several years, and I would expect something at the same pace of adoption to occur in the microbial arena as well.
Well, that seems like a great place to finish. This has been a really interesting conversation, and I'd like to thank all of our guests for sharing their insights into the development of this unique solution. I'd also like to thank you, our listeners. If you'd like to learn more about Gibco Bacto CD Supreme fermentation production media, please visit thermofisher.com/CD Bacto Supreme or reach out to a Thermo Fisher scientific representative.